Okay, welcome to the next lecture on plant diversity. After the previous lecture where we talked about different plants and their adaptations to different microbiomes, we're going to go back in time now and look at the evolution of plants from their algal ancestors through non-vascular plants and then seedless vascular plants, and look at how life cycles also have changed over those through that evolution. So learning objectives, remember that's kind of your study guide for this lecture, what you need to know and what you need to be able to recall. And then vocabulary words, these have strong overlap with vocabulary that you'll be covering also in the lab. Okay, so as I said, let's go back in time and look at geological time scales. So along the x-axis here, we've got time. Current time is right here on the right, and then we go back in time, back in giga years, that's 1 billion years before the present. So, and then on the y-axis here, we've got relative oxygen concentration in the atmosphere relative to what it is now. So if it's 100% now, what was it at a particular time going back in time? So you can see that in the early period of the Earth, for a very long time period in the Earth, there was virtually no oxygen in the atmosphere. And then we had the evolution of cyanobacteria. You may remember that from the microbiology lectures. These are bacteria that have the ability to do what we call oxygenic photosynthesis. That's using light energy to fix carbon, and they produce oxygen as a byproduct of that. There's other bacteria that do different types of photosynthesis which doesn't produce oxygen. But cyanobacteria were really critical here. So slowly over evolutionary timescales, those cyanobacteria started to produce oxygen, and you see oxygen starting to build up a teeny little bit here. And then by contrast, a lot more recently, we had the evolution of the eukaryotes. These are those organisms that have a nucleus and other organelles inside. So, and then the cyanobacteria gave rise to the evolution of green algae through the endosymbiosis. And so you have chloroplasts inside these green algae that are also doing photosynthesis. And you can see that the oxygen concentration is starting to rise. And then you have a whole range of other things that were starting to evolve and appear on the Earth through evolution over those time periods. And then the oxygen concentration started to go up. And land plants started to arise around here, around 450 million years ago, when the green algal ancestors started to move onto land. And of course, land plants also were doing photosynthesis. So now we have photosynthesis going on both in the oceans and water, but also on terrestrial ecosystems, and that caused the oxygen concentration in the atmosphere being churned out through photosynthesis to increase even more. And then of course sometime after that we had the evolution of animals. Okay, so I'm going to go through a series of slides here showing you a little bit of the prehistory of land plants, and this is mostly based on what we see from fossils. And then on the right hand side here we have a geological timescale. Maybe some of you are taking a geology course at UWM. If you are, I hope that's really useful for you to put these in context of where and when did land plants evolve. Okay. So the Precambrian period, we describe that as that's about greater than 550 million years ago. And what was happening during that time? That's when we got those cyanobacteria first evolving, and the first photosynthesis that was producing oxygen. That was about 3,000 million years ago, or 3 billion years ago, and that's when we started to get very slowly the increase in oxygen in the atmosphere. And then we had that endosymbiosis events occurring, leading to eukaryotes, cyanobacteria, bacteria. We had primitive protists, which are also able to do photosynthesis. This probably occurred about 1,500 million years ago. And then after that, we started to get the evolution of plants during the Precambrian into the Paleozoic period, which is shown here in Paleozoic is here in blue. So what did we see then? We saw that algal protists, these organisms that can do photosynthesis that are growing in oceans and freshwaters, they started to diversify. That means that you get more and more new different species evolving around 1,000 to 600 million years ago. And then the first green algae appeared around 750 to 700 million years ago. And the first plants, by contrast, were much more recently, about 490 to 438 million years ago. So that's going to be kind of in this period here, the Silurian Devonian periods. So land and plants started to colonize land about 470 million years ago, also during that period. By contrast, the first terrestrial vertebrates didn't start evolving until a little bit later here in the late Devonian period. So that sort of puts that into context a little bit. So now we're also, when we're talking about evolution, we really need to talk about phylogenetic relationships. And you've hopefully encountered this in the lab and also in the microbiology section. So let's unpack this. Phylogeny means, comes from the Greek philos or tribe, and genesis, of course, meaning origin. So where did the different tribes or groups originate? How did they originate from each other? So this really describes evolutionary origins based on a whole range of different characteristics that we think we can see similarities between groups. This used to be purely fossil evidences and anatomical similarities. Increasingly, we're using molecular or gene sequences to determine relationships and relatedness between organisms. So a phylogenetic tree is like a family tree showing how things are related to each other, and this is estimating their evolutionary relationships. And I will add that amongst taxonomists and evolutionary biologists, these trees do change a little bit. People are often arguing about, no, I think this group is more closely related to this group, etc. So this thing, while we have a really big picture things worked out, they do shift. Some of the smaller little branches on those trees are moving around a little bit. So here's a phylogenetic tree of the group Plantae, which is the plants, including some of the algae, which are the closest ancestors, and of course all the way up here to the land plants, sometimes called the embryophytes. So you can see here, these are some of algal ancestors, the protists that you saw, we'll see in the lab, and then here we have green algae, and a range of different green algae, and then some green algae that we think are even more closely related to plants, including the chara, this one here, the charophytes, which are a type of green algae, but that's one that's particularly closely related to plants. We also have Coleochaete here, which is also regarded to be quite similar to higher plants. So in each one of these little purple reddish dots shows you a particular evolution of something in common that everything more recent than that has. So here we have things like cuticle development and a multicellular sporophyte. So we'll come to some of these evolutionary characteristics that we can see over time. And so here are the land plants, the plants that have now become terrestrial. And they've evolved from these charophytes. Okay, so here we have some green algae and those chara and Coleochaete, which are believed to be the most recent living ancestors of higher plants. So this freshwater green alga chara is often regarded as the direct ancestor. Here you can see it growing kind of in the water, and here's a close-up, and we'll also look at that in some detail in the lab. So when we look at land plants and their green algal ancestors, there's certain things that we can say that they have in common, which tells us that they're kind of related. So key features that are shared by land plants and their algal ancestors. These include a cellulose cell wall. Some other further back algae have different compounds that they use to make cell walls. They also use starch as a carbohydrate form to store energy. And they have similar photosynthetic pigments. So chlorophylls are those green pigments that make plants green, chlorophylls A and B. But then we also have other pigments called carotenes, beta carotene, which is responsible for making things orange like carrots, but also in petals of flowers here. These are shared in between algae and the plants that evolve from them. We also see similarities in the way they do cell division and mitosis and cytokinesis, so that's the cell dividing cells. And also within reproduction, we see similarities in sperm ultrastructure. So algae reproduce by sperm that have to swim, and in some cases in plants we also see similar structures embedded in motile sperm. Also, we see a lot of gene sequence similarity. We often call this molecular data. It means that we sequence the genes that do similar things in these and line them up together and say, yeah, they look pretty similar. There's just some little differences in this sequence. And then also in life cycles, and we're going to talk about life cycles a bit more. So on Canvas, we'll have a discussion about what are some of the challenges of moving onto land. If you have an algal ancestor and then it has to move onto land, what's going to be the challenges? So look out for that on Canvas. So when we're talking about evolution of land plants, so transitioning to the land, one of the first things that is going to be important is to protect against loss of water, so desiccation resistance. We also have to anchor ourselves somewhere. So if you're a plant in the water, you might be able to float around, but that's not going to be so easy on land. You might get blown away by the wind. And so root-like rhizoids started to evolve. And these provided some structural support, but also anchoring them to the substratum. So the arrow over here on the right is showing over time what is the order in which some of these adaptations evolved. Then we have primitive vasculature, or ability to move water around in the plant, and really simple primitive leaves started to evolve. And if you think about a plant also on land, it has an aerial component that's in the air, and so it needs some sort of structural rigidity or some support mechanism to support itself in air. If you think about the blue whale that is supported by the water in the ocean, if you bring it out on land, it quickly will die because it's not supported well enough and it crushes itself. So plants aren't that big, but it's a similar idea, and they use a compound called lignin, also in woody tissue, to help give them structural rigidity. If you have a cuticle over the surface of the leaves to prevent water loss, you still need to get gases in and out. So stomata are little pores inside in the cuticle that go from the outside into the middle of the leaf to allow gases to come in and out of the leaf. So we can see this in the prehistory of plants plotted through fossil evidence. Early land plants, you can see fragments of cuticle, which is one of the first things that arose, and this fragment is believed to come from around 450 million years ago. We see seed fern leaves from very early time period during the Carboniferous, cones from an early gymnosperm, and an early angiosperm. So over time, you're getting different and new innovations or new adaptations to allow plants to grow even better on land. So these key adaptive innovations are shown in this figure here. So for example, here are stomata. Before that would have been cuticles. And we have things like tracheids, vascular cells. These are the first cells that were important for being able to conduct water, which is really important because if a plant is growing in the air, it needs to get water through its roots from the soil. And so it needs a way to move the water around. We have megaphylls, we have heterospory, and we have flowers and a triploid endosperm to get to the angiosperms. So you're going to do an exercise in the lab about this, looking at the evolution of different components. And the other thing that we can think about with these adaptations is many of them, they're all moving towards increasing desiccation tolerance. So being able to live in drier and drier ecosystems on the planet after those early land plants that have evolved for living on land, but they're still dependent on needing a fairly moist environment. And so we're going to talk about that a little bit later in this lecture. Okay, so now I'll come to life cycles. And life cycles are important because they tell us a little bit about evolution. And just a quick revision, mitosis and meiosis, if this you're going, oh, I can't remember which they are, just have a quick revision on this. I remember it by mitosis produces mito, which is just more of me, more new cells, which are the same as the rest of my foot. Whereas to get me, a whole new organism needed meiosis to produce gametes in my parents, which came together to produce a zygote, which grew into me eventually. Okay, so there's 3 different life cycles that we're going to encounter in plants, and these overlap with some of the ones that we looked at in fungi as well. So haploid dominant means that the gametophyte stage, or the haploid component, is dominant, is the biggest, most obvious part of the life cycle. That if you're looking at a plant, you'll see it. Whereas in this case, we have a diploid cell, very minimal diploid phase, and that's why it would be haploid dominant. So an example of this is mosses. We're going to come back to this in a little bit more detail in a moment. So the next, and so yes, so that the diploid phase is very, very tiny in this case. So the diploid dominant is probably more familiar to you. This is most kind of really big plants that you can see, trees and garden plants that you may be familiar with. So this is diploid dominant, which means that the adult plant that you can see here is a sporophyte, it is diploid, and the haploid component of the life cycle is actually microscopic, and it's inside or within the parent. So this occurs both in gymnosperms and in angiosperms, which we'll cover in the next couple of lectures. And then the third life cycle type that we're going to have is kind of a codominant, sometimes also called alternation of generation. So we have a haploid gametophyte here, which is photosynthetic, it's independent, it is fairly small, but it is still macroscopic. And then we have also a macroscopic and independent and photosynthetic sporophyte stage. So that's diploid. So we say that these are kind of coexisting. Both generations are macroscopic, which is what the definition really is of that alternation of generation. So we're also now going to move on to looking a little bit of these non-vascular plants. These were the earliest land plants that evolved, and they include liverworts, mosses, and hornworts. So together, these 3 groups are called the bryophytes, and these are non-vascular plants. Whilst they live on land, they haven't yet evolved a true vascular system. So they live in moist and wet habitats because they lack true leaves and lack true ability to be able to move water around within the plant very effectively because they don't have vascular tissue. Liverworts and hornworts are totally non-vascular. Mosses have some really limited vascular cells, just sort of indicating kind of towards a vascular plant evolution. And they also need water to complete the life cycle. We'll see why in just a little bit. They don't have any woody tissue. They're very kind of flat and small, and so they haven't been able to get big because they don't have the support that they would need on land to get big. So mosses, probably the most familiar, over 12,000 species. They grow together in these kind of dense mats, and this is about helping retain water within the clumps. And and they have just rhizoids. They don't have real roots that are attaching into soil. They just attach very gently to the substratum. So if you see some moss, you can almost put your hands underneath and peel it up off the rock or anything it's growing on. They have a stem and leaf-like structures, but they're not very well developed, and they're very, very tiny, very small. So the life cycle that you can see in this picture here, that is a gametophyte. That's the noticeable green gametophyte. So it's a gametophyte dominant phase. So the diploid part of the phase occurs when we have the gametophytes here producing gametes, and then these come together in fertilization, and then they produce this diploid sporophyte phase, which is attached to and dependent upon the gametophyte. So the diploid capsule here is a capsule of diploid tissue, which then inside that undergoes meiosis to take the haploidy level from diploid back to haploid to produce haploid spores. The haploid spores can be dispersed out and then grow into a new gametophyte. So here we are with the whole life cycle here. We start here with this haploid gametophyte, the green phase. On the surface of that, you have Antheridia and Archegonia. So the Archegonia is the female gamete producing structure. So it has an egg inside here. And then the Antheridia is the male gamete producing structure. So it's a little bit like anthers that you find in flowering plants. That's where the name comes from. But actually the male gametes here are motile. They are little sperm, and they have to swim out of the Antheridia here through some water to get down into the archegonium to fertilize the egg. So we have fertilization occurring now in this life cycle inside the archegonium, and now we have a diploid zygote, and then that grows into a diploid embryo, and that grows out of this structure here to form this little spike with a capsule on the end, and then inside that capsule you have meiosis going on to produce haploid spores, which then get dispersed. Each spore can germinate and grow into a new gametophyte here, which eventually grows up to form this gametophyte, which can produce Antheridia and Archegonia again. The hepatophyta, the liverworts, are another group of the bryophytes. You may have seen these in kind of wet forested areas. 6,500 species. It's sort of like a filmy kind of layer or thallus over the surface of the soil. It has the same life cycle to mosses, except it also produces asexually from these little gemmae and gemmae cups. And we may be able to show you these in the lab materials. They also reproduce sexually. So here you can see the sporophyte, the diploid components, which are going to then meiosis going on inside, and then releasing haploid spores again. The Anthocera or hornworts are a little bit similar to liverworts. They have slightly different structure, and that they're a different group. Only about 100 species, similar in appearance to liverworts, and the sporophytes, instead of being the capsule on the end, they just have these sort of elongated capsules, but this is the diploid component that's going to undergo meiosis to produce spores. So bryophytes are restricted to moist habitats. Think a kind of, a rainforest in the West Coast, where you've got trees dripping with mosses here. So wet habitats, If you think about the gametophyte here, it's only got tiny little rhizoids. It doesn't have proper roots. So taking up water from its surroundings is going to be difficult. It doesn't have good roots to do that. So it's going to need to be fairly close to water to get enough water to maintain itself. The other really important component, of course, is those swimming sperm. Sperm can't swim unless there's water, so you're going to have water around for them to be able to complete that life cycle. So these were the bryophytes, the most primitive of the land plants, the earliest land plants that have the least adaptations to a dry ecosystem. And then next we're going to move on to the, after the evolution of true vascular tissue here, we're going to move on to the seedless vascular plants here. And this will be in the next part of this lecture. Increasing desiccation tolerance. So life cycles are something that are going to come up in the next few lectures and in the fungi lecture and of course in the lab. So I'm going to leave you with a little break here where you can think about this generalized life cycle and practice thinking about this. This is an active learning that I talked about before. This is, if you can make it active, it's going to be more valuable. And think about how you fill in the missing terms. So what would you need to put in this A box here? So have a go at that and I'll see you shortly.